So first second. Okay. So welcome to now NGS online workshop. You can find the syllabus at bit.ly/NGS_analysis. You can also register online with your name, email and mobile number at bit.ly/register NGS or in NGS in caps. You can also subscribe, save, share and hit the bell icon to YouTube channel bit.ly/YouTube-bio-info and join NGS_analysis WhatsApp group at bit.ly/NGS and contact NGS Instructor at this number, myself, and connect on LinkedIn. Okay, so we were in the last section of the variant annotation, right? So let us go to 40. We completed this multi-sample variant column. So why is variant calling challenging? We discussed yesterday. And why is it so difficult to call variants? Did anybody join Google Meet? Let's check. Brajeshi, okay. So we'll check. Is Brajeshi screen visible and am I audible? You can reply in chat. Yes, okay. So then why is it so difficult to call variants? Yes, can anybody read this?
Okay. The most genomic related DNA sequences have several properties that confound institution as they require thinking about the context that human mind does not typically need to solve. Number one, the DNA sequences can be exactly long, million of the bases. Number two, just our building blocks ATGC make up any DNA sequences. These, even when distributed randomly, could provide a variety of patterns just by the sheer chance. The DNA only partially affected by the randomness. Usually, most of it has the function. Depending on the organism, various segments of the DNA are affected by the different rate of randomness. The experimental protocol break long piece of DNA into tiny ones that we need to attempt match back a reference. Ideally, we hope that it is still match the most closely to original location, because only then will our match be a variant of the site of interest.
Okay. So basically the
Our
DNA sequence is exceedingly long, millions of bases. So when doing the SNP calling, uh, the separation between the genomic positions, that is quite important, right? So separation between the genomic positions will give us something like estimate of the variation density, SNP density. If there is less separation, we can say that it is more dense. If there is more separation, it is more sparse. So just 4 building blocks ATGC make up any DNA sequence. So at every position we have 4 possibilities, right? So as the positions are going further from 5 prime to 3 prime, so exponentially the randomness grows. So first position 4 power 1, second position 4 power 2, third position 4 power 3. So at millionth position 4 power million, right? So randomly, depending on the length, exponentially randomness is growing. DNA is only partially affected by randomness. Usually, most of it has function. So, regulatory genomics we know, right? So, most of we know a gene. Gene is basically a region. There are many algorithms to predict gene. Also, that's not completely perfect. So, there are regulatory regions in the genome which have function. So, depending on organisms, various segments of DNA are affected by different rates of randomness. So, for example, most of the regions in the case of comparative genomics, they are conserved across species, but for some, depending on the genome size, those regions may be more compact. So different rates of randomness, it depends on how the evolution has taken place. And experimental protocols break long pieces of DNA into tiny ones if in order to match back to a reference. So we saw in a previous section that while doing the indexing of the genome, we take particular multiple segments of the reference and then do an indexing process. Ideally, we hope that it still matches most closely to the original location, because only then will match the variant site to be of interest. So here, what are we doing? We are not doing mapping, right? We are doing alignment. So alignment is important for SNP calling, and mapping is important in case of RNA-seq. Okay, so how do variant calls overcome these challenges? So the process has different implementations can be realignment or probabilistic alignment. So when sequences are short, each is affected slightly differently. It turns out it's possible to attempt to recover the correct alignment from all the different types of errors that affect each shorter read differently. It is feasible to find another variation that will better explain and reconcile the differences present in each read. So how to explore the effect of mutations on variant callers? Okay, we can compare variant callers, SAM tools, FreeBayes and GATK. So let us see the scripts. Obviously this will require the installation, so let us check once. Okay, so let us first download the script and then check whether the GATK is used from on basis of Java archive or on basis of Bioconda GATK. We have 3 tools. Okay, so I'm doing CD NGS 07. This is a small script for comparing variant callers. Already we compared variant callers in the last section. We took FreeBayes, we took VCF tools. We saw that the genomic positions for variants are changing. We'll just wait for 1 minute. If this is not downloading, you can see that we already compared variants in last section. Okay, so next basically 3 VCF files are generated: samtools.vcf, freebayes.vcf and gatk.vcf. Okay, so there is the file called genome.fa contains the genome from which the sequencing samples are generated. With the method of our choice, we can modify and save this file. We will use the command line to biosave to create a SNP replacing a with a T. We also have selected this variation to be at position 2000 to help us more quickly identify whether the SNP caller indicates it correctly. So so fundamental thing is to get this script only. That's the first thing. Or if we not able to get the script, we see that we already compared 2 variants yesterday, 2 variant callers yesterday. So it should be expected that different variant callers produce different VCF files. Okay, so this script, let us check one last time here. Well, just go to this link itself, up to this link. Okay, let it be retrieved. We can go to next section, because comparing variants is not new to us. We just compared free bayes and BCF tools yesterday. Variant normalization. Yes, Shipraji, can you read this?
A genetic variant can be represented in the variant call format VCF in multiple different ways. Inconsistent representation of variant between variant callers and analyzers will magnify discrepancy between them and complicate variant filtering and duplicate removal.
Yes, so for example, the same variant could be represented as in this way. So GAC becoming AC, what is this?
Two different variant callers produces two different variants.
That is
So
fine.
there is a
GAC
discrepancy
becoming AC, what is it?
deletion.
Deletion, right? So TGAC becoming AC, that is also deletion, right? Yes.
Yes.
Yes. So GACC becoming ACC, this is also deletion. So there is no universal thing applying here. So variant normalization is a process of simplifying the representation of a variant to obey the following rules. What are the three rules?
Represent the variant with as few letters as possible. Number 2, no allele may be of B. Number 3, variant must be left aligned. This also applies to repeated regions.
Okay, what is left aligned? A variant is left aligned if it is no longer possible to shift its position to the left while keeping the length of its allele constant. For example, let us take CCCC would need to be reported at the leftmost position. So only C, right? So a variant is left aligned if it is no longer possible to shift its position to the left while keeping the length of its alleles constant. For example, insertion of C into CCCC would need to be reported at the leftmost position. So if you insert a C into this, the constant length is changing, right, from 4 to 5. So we need to keep it at the leftmost position so that this insertion will be with as few letters as possible. Should variant callers report normalized variants by default S, and eventually they will. GATK, for example, already produces normalized variants. The output of other tools needs to be normalized. How do I normalize a variant? So this is the subtool BCF tools norm qualifier ref for $ref and samples.bcf. Okay, yesterday itself we generated multi-sample variants, right? So we can do that. BCF tools norm. So if you go back to that folder, so this clear cd NGS 06 October. So we have 4 VCF files. Let us check how many VCF files we are having. ls qualifier lha star.vcf. This is the last 4 VCF files we are interested in. So let us then compare just BCF tools and free bayes. So we will do this grep qualifier v single quote caret hash BCF tools dot VCF and head qualifier n4 okay n5, and then similarly we will do free bayes dot VCF, right? So these are the first 5 variants for BCF tools and free bayes. So GCG is CCG, and let us normalize the variance. Let us check what is ls, and then refs is there, right? So again let us assign the variable, no problem. So ref, let us check it, echo $ref. Yes, it's not there. So we have assigned the variable again. So cd ref, just let's check it, af086833.fa, right? So let us copy this, cd double dot ref is equal to, what should be here, yes, this is a reference. Okay, so let us do the tool VCF tools norm qualifier f $ref, and then let us do first on VCF tools.VCF. Yes, so now it's normalized. So now we see that almost all are single characters, right? As few letters as possible, the variants are represented in. So this will become more clear when we do this for this. First one, let us check VCF tools. There's only one letter in the first variant position, 127. For free bias.VCF, we have three bases, right? GCG changing to CCG. So now let us normalize this and then check back. So VCF tools norm space qualifier f space $ref space free bias.VCF. Yes, let us check it. So it's now at 44th position, only G is changing to Chi, right? Only one character here, right, at 44th position. Can you see that? Yes, can you see this? Hello?
Yes, we
Yeah,
can see this.
yeah. So previously it was, if you see it is GCG changing to CCG. So CG is common, right? CG is common. Yes. CG is common. G is the base that's altering is G to C, right? So this will be represented in as few letters as possible, G to C. Okay, so now it is normalized. So clearly variant normalization may be more significant for this session. Okay, so then VT normalize qualifier f $ref samples.VCF. This is also one more way we can do the normalization. So how do we normalize VT normalize qualifier $ref samples.VCF? Okay, so let us do this separately, right? Let us check it again, free bias. So now GCG is going to CCG. Basically the original file is not altered. When you do the variant normalization, the output which comes, you can store it if you want in a new VCF file. Okay, the original VCF file will remain as it is. Okay, let us do this VT normalize. Anyway, it's not required to do for VCF tools.VCF because it's already containing one variant in that position. Much where normalization may not be done there. So free bias.VCF. So this is giving additional information, right? No left trim 0, no right trim 1, left and right trim 0, right trim then left align 0, left align number left align 0, total number of biallelic normalized point, and multi-allelic for biallelic, multi-allelic, and total number of multi-allelic normalized. Okay, so if you check this again, it should give expected same output only, like BCF tools normal. So if we go up at 44th position, right? Yes, again it is single base, right? G to C, right? Yes, G to C, right? Yes, again it is represented in as few letters as possible, single base resolution, guanine changing to cytosine, right? Yes, hello. Again it is just represented as G to C. Yes.
Yes.
Okay. So we can store if you want this in new VCF file. That's not a problem. You can just do output redirection if you want. So there are two tools for variant normalization: BCF tools norm and VT space normalize. This is what we did, right? VT space normalize and BCF tools norm. Okay, so then let us go further. Variant annotation and effect prediction. Okay, variant annotation means predicting the effects of genetic variants, single nucleotide polymorphisms, insertions, deletions, copy number variations, or structural variations on the function of genes, transcripts, and protein sequence, as well as regulatory regions. Variant effect annotators typically require additional information from other data sources. The predicted results will depend on the type of other data that is known about the organism or genome under study. The starting points for variant annotator or variant sets, insertions, deletions, multi-nucleotide polymorphisms, usually in VCF format. The result of variant annotation may be reported in text, HTML, or PDF format, along with an annotated VCF file that now contains additional information on each variant. The text or HTML reports contain substantially more information than the VCF files, as this latter format limits the type of content that may be added. Okay, so what are the consequences of variant effects? Whereas the way the word is used seems to imply a universal effect, let us keep in mind that all effects are related to the information that's currently present in the database. So recently you would have seen, for example, that SNPF, if you check, COVID-19 variation is also included in the annotation. If you check carefully, SNPF bioconda. So if you see this in bioconda, you can see this, right? 4.5 COVID-19 1, 4.5 COVID-19 0.4.3. Yes. Yes, you can see the versions, right? Hello.
Yes.
Yeah. So recent annotations of COVID-19 has also been added to the SNPF. I think we installed SNPF last time. Let's just check SNPF. Command not found. Okay, so we will install this. But before that, let us first check what are the tools being used here. SNPF is used, yes, and then SNPF, okay, it is, and then I think VEP, variant effect predictor, is also used.
Main or 10 minutes off here, main.
Okay. Okay, so there's electric work